Imagine trying to fix a sputtering jet engine while it's literally running at full speed.
Right. And you're entirely blindfolded.
Exactly. You're blindfolded, and the only way you can figure out what's wrong is by smelling the exhaust fumes.
It sounds completely impossible. You're, entirely removed from the physical mechanics of the problem.
But that is essentially what internal medicine is. I mean, when a car engine breaks, a mechanic pops the hood and points to a snapped belt. But when you step into the world of human biology, that hood is welded shut.
Yeah, the moving parts are entirely microscopic. The chaos is happening on, well, a molecular level.
Which is exactly why we are taking this deep dive today. So, welcome to the show. Whether you are a medical student prepping for boards, a young healthcare professional on the wards, or just, an insanely curious learner, you are in the right place.
Absolutely. Our mission today is to synthesize the key concepts from Mark's basic medical biochemistry.
Right. We are going to provide you with a really comprehensive, clear, and hopefully highly engaging summary of how the human body converts food into energy, how proteins and enzymes regulate life, and, this is the cool part, how doctors use those invisible biochemical clues to diagnose disease.
We're basically treating this dense medical text as a fascinating instruction manual for the human body. And the goal isn't just to memorize facts. We want to explore the what, but more importantly, the how and the why behind these complex systems.
To pull that off, we're going to use clear scientific language, avoid unnecessary jargon, or at least, explain it when we really have to use it, and connect theoretical biochemistry to very real clinical examples.
Oh, and make sure you stick around because there will be a special review question at the very end of this deep dive to test your knowledge.
Yes, definitely stick around for that. So before we can talk about disease and diagnostics, we have to understand how the body's engine actually runs on a good day.
Right. And that begins with the most fundamental requirement for life, which is energy.
Okay, let's unpack this. Everything we eat gets processed through metabolic pathways, which really boil down to two main directions, right?
Exactly. Two directions.
You have catabolic pathways, which are like the demolition crews. They break down larger molecules, like, say, the fats and carbs in your lunch, into smaller components to release energy. And then you have anabolic pathways.
Which are the construction crews. They build things up, like synthesizing glycogen to store energy for later.
Right. And the ultimate goal of those catabolic demolition-style pathways is to generate the body's primary energy currency.
ATP, adenosine triphosphate.
The big one.
Yeah, the body does this through cellular respiration, which happens in two distinct phases. Phase 1 is the oxidation of our fuels.
Meaning glucose from carbs, fatty acids from fats, amino acids from proteins.
Exactly. They all get funneled down and oxidized into a crucial central molecule called acetyl-CoA.
It's almost like all the different currencies of the world being exchanged into one universal dollar before it goes into the bank.
I like that. That universal dollar, acetyl-CoA, then enters the tricarboxylic acid cycle. Most people know it as the TCA cycle or the Krebs cycle.
Oh, right, the Krebs cycle, the bane of every undergrad's existence.
Right. But here, it gets completely oxidized into carbon dioxide. But the real magic of the TCA cycle is that it produces these specialized electron-carrying coenzymes, specifically NADH and FAD2H.
I always picture NADH and FAD2H as little armored shuttle buses. Their entire job is to just pick up high-energy electrons from the broken-down food and transport them to the next phase.
And that brings us to phase 2. Those shuttle buses head over to the electron transport chain, which sits inside our mitochondria.
The powerhouse of the cell.
Exactly. They drop off their electrons, which get passed down a chain until they're finally handed off to molecular oxygen. And that final handoff is literally the reason you and I breathe oxygen.
Wow.
Yeah. The massive amount of energy released during that electron transfer is harnessed to convert depleted ADP back into fully charged ATP. We call this oxidative phosphorylation.
It really makes me think of ATP as a rechargeable battery. When the body does, biochemical work like a muscle contracting or the liver detoxifying a chemical, it uses up that battery, snapping off a phosphate group and leaving behind ADP.
Right. And then the electron transport chain recharges it.
But wait, what about getting energy straight from sugar? Because I know the glycolysis pathway is a major player there. How efficient is that process?
Well, glycolysis is highly conserved across almost all life forms. You take one 6-carbon molecule of glucose, and the cell splits it into 2 3-carbon molecules of pyruvate.
Okay.
By breaking those chemical bonds, the cell nets 2 ATP batteries, fills up 2 of those NADH shuttle buses, and releases 4 hydrogen ions.
So it's pretty solid yield.
It is. And what's really efficient is that other dietary sugars, like fructose from fruit or galactose from dairy, they feed right into this exact same biochemical highway.
Oh, that's smart. The body doesn't need to build a whole new highway for every single type of sugar. It just merges them onto the main road.
Exactly. And the traffic on that road is heavily regulated based on whether you are in a fed state or a fasting state.
Right, because when you eat, your pancreas releases insulin.
Yes, insulin is the fed state signal. It tells the body's cells to open their doors, use the glucose for immediate fuel, and store any excess as fat and glycogen for later.
But when you haven't eaten in a while, the pancreas releases a different hormone, right?
Glucagon.
Glucagon, yeah, that's the fasting signal. It basically sounds the alarm, telling the liver to release its stored glucose into the blood and telling adipose tissue to release stored fatty acids.
So the body doesn't starve.
Exactly.
But I have a question about this whole oxygen-dependent system. If aerobic respiration and oxidative phosphorylation are so incredibly good at making massive amounts of ATP, why does the body even bother with anaerobic pathways?
You mean making energy without oxygen?
Yeah. If glycolysis is so efficient, why do we have this other pathway? It really comes down to cellular survival. Anaerobic glycolysis is a crucial backup system. When oxygen isn't available, the electron transport chain completely shuts down.
Oh, because there's no oxygen to catch the electrons at the end.
Exactly. If a cell can't run that chain, it diverts the pyruvate from glycolysis and turns it into lactate instead.
Which is why our muscles burn when we work out too hard.
Yes, we see this during intense exercise, when your muscles are consuming oxygen way faster than your blood can deliver it. We also see it in red blood cells, which literally lack the mitochondria needed to use oxygen in the first place.
Wait, really? Red blood cells don't have mitochondria.
They don't. So by converting pyruvate to lactate, the cell can keep generating a tiny, steady stream of ATP. Without that backup generator, the cell would rapidly die the second oxygen levels dropped.
That makes perfect sense. It's a biological failsafe. Okay, so now we understand how the energy is made, but we also need to understand the underlying rules of the universe that govern these reactions.
You were talking about bioenergetics.
Yes, because every single chemical reaction in the human body follows the strict laws of thermodynamics. In biochemistry, we measure this using Gibbs free energy, which is denoted as delta G0 prime.
Right.
And I remember looking at that equation and thinking, wait, do doctors actually calculate Gibbs free energy at a patient's bedside? Like, are they doing thermodynamics math in the emergency room?
What's fascinating here is that while nobody is crunching those specific numbers on a whiteboard in the ICU, the core concept is essential for diagnosis.
How so?
Let me explain it without the math. If the delta G is a negative number, the reaction is exergonic. Think of it like a boulder rolling down a hill. It happens spontaneously and it releases energy. Oxidizing your food is exergonic.
Okay.
But if the delta G is positive, the reaction is endergonic. That's like pushing the boulder up the hill. It requires an active input of energy. Building muscle is energonic.
So the body basically uses the energy released from the boulder rolling downhill to power the process of pushing other things uphill.
Exactly. And clinically, this explains a lot of pathologies. Consider a patient named Stanley T., who suffers from hyperthyroidism.
Okay, so his thyroid is overactive.
Right. Thyroid hormones normally set our basal metabolic rate. But in hyperthyroidism, excessive thyroid hormones dramatically accelerate cellular processes that consume ATP, like the sodium-potassium pumps in our cell membranes.
So his body is constantly forcing that boulder up the hill, hydrolyzing massive amounts of ATP.
Yes. And because no energy transfer is perfectly efficient, a lot of the energy from those exergonic reactions is lost as heat.
Oh, wow.
That underlying thermodynamic principle is exactly how a doctor knows why Stanley T is presenting with profound weight loss and excessive sweating. He's quite literally burning up his fuel stores.
Man, thermodynamics is one rule, but there's another major environmental factor inside the cell, right? pH.
Absolutely. Our blood has to maintain a very delicate pH balance, hovering right around 7.4. If it gets too acidic or too alkaline, proteins just stop working.
And the body manages this using chemical buffers, which are governed by something called the Henderson-Hasselbalch equation.
Right. And rather than getting bogged down in the algebra, think of the Henderson-Hasselbalch equation as a mathematical scale that balances an acid against its conjugate base.
Okay.
It helps us understand how a buffer system like the bicarbonate in our blood soaks up excess acid to keep the pH stable.
But when that buffer system gets overwhelmed, it's a massive crisis.
A textbook example is diabetic ketoacidosis, or DKA, We see this in patients with type 1 diabetes. These patients do not produce insulin.
And if you recall, insulin is the Fed's signal.
Exactly. Without it, the body literally believes it is starving to death, even if there's a massive amount of glucose floating around in the blood. The cells just can't access it.
So the liver panics and starts rapidly breaking down fatty acids to create an emergency alternative fuel source.
Yes, those alternative fuels are called ketone bodies, specifically acetoacetic acid and beta-hydroxybutyric acid. And the problem is right there in the name. They are acids. As the liver dumps massive quantities of these ketone bodies into the blood, they dissociate and release hydrogen ions.
Which instantly tips that Henderson-Hasselbalch scale. The blood's bicarbonate buffers try to soak up the acid, but they just get completely overwhelmed.
And the blood pH plummets, resulting in a life-threatening metabolic acidosis. The environment turns toxic.
Wow. Okay, so we've established the energy pathways and the delicate operating environment. But what is actually doing the physical labor inside the cell to make all of this happen?
That brings us to the physical architecture of life, proteins.
We're moving from the fuel to the actual machines. And to understand the machines, we have to look at the blueprints.
The central dogma of molecular biology.
Yes. DNA sits in the nucleus and is replicated. Then it's transcribed into a working copy called RNA, specifically messenger RNA, transfer RNA, and ribosomal RNA.
Right.
Finally, that RNA is translated into proteins on the ribosomes.
And the translation process is remarkable. The ribosome reads the messenger RNA in sets of 3 nucleotides, which we call codons. The process almost always starts with the codon AUG, which codes for the amino acid methionine.
Right. AUG is the start signal.
Exactly. From there, it strings together a long linear chain of amino acids.
And that linear chain is the primary structure of a protein. But a protein isn't just a floppy string. It folds. It's like origami.
Origami is a great way to put it.
If you are folding a complex paper swan, the final shape depends entirely on making the right creases in the right order.
And the folding happens in stages. That primary sequence dictates the secondary structure, forming patterns like alpha helices or beta sheets, which are locked in place by hydrogen bonds.
Okay.
Then it folds even further into a tertiary structure, a complex 3D shape. In aqueous environments like our cells, the protein usually folds so that its water-hating hydrophobic amino acids are hidden in the core, while the water-loving ones face the outside.
Thanks.
Some proteins even have a quaternary structure, where multiple fully folded subunits lock together to form a supermachine.
And the fragility of this origami is terrifying when you look at it clinically. I mean, take sickle cell anemia. It is a devastating disease, and it is caused by a single microscopic typo in that primary structure.
We call it the E6V mutation.
E6V.
Yeah. At position 6 on the primary sequence of the hemoglobin beta chain, the DNA blueprint accidentally swaps out the amino acid glutamate and replaces it with valine.
And glutamate likes water, but valine is hydrophobic?
Exactly. It's one wrong crease in the paper swan. Because valine hates water, it creates a sticky hydrophobic patch on the outside of the folded hemoglobin protein.
Oh, wow.
So when the cell drops off oxygen, these sticky patches latch onto each other. The hemoglobin molecules clump together into long, rigid fibers.
And those fibers physically distort the red blood cell, stretching it into a rigid sickle shape.
Yes. These sickled cells can't bend, so they cause microscopic blockages in the blood vessels, leading to immense pain and tissue damage. Form entirely dictates function.
That is wild. And speaking of form, I want to ask about tissue-specific isozymes.
Okay.
I know these are proteins that do the exact same biochemical job, but they have slightly different structures depending on where they live in the body.
Right, like creatine kinase or CK.
Yes.
No.
You have the CKMM version in skeletal muscle, the CKBB version in the brain, and the CKMB version in the heart. But hold on. If my DNA blueprint is exactly the same in every single cell of my body, how does my bicep know to build CKMM while my heart knows to build CKMB?
This is the elegance of tissue-specific gene expression. Even though every cell holds the entire DNA library, they don't read every book. Different cells contain different transcription factors, specialized proteins that tell the nucleus which specific chapters or genes to transcribe. The heart's transcription factors turn on the CKMB gene, while the muscle's factors turn on the CKMM gene.
And clinically, that is a massive diagnostic cheat code for doctors.
It really is.
Because if a patient comes into the ER and their blood tests show sky-high levels of the CKMM isozyme, the doctor knows immediately that skeletal muscle is being destroyed. But if it's CKMB, the damage is localized specifically to the heart.
Exactly. It allows us to pinpoint the invisible damage. Now, proteins don't just sit around as structural scaffolding. Some of them act as enzymes.
Right.
These are the active catalysts that drive every single metabolic pathway we've discussed so far.
Enzymes are the ultimate efficiency hackers. They speed up chemical reactions without being consumed themselves.
Yeah.
They have a specialized pocket called an active site where the target molecules or substrates bind perfectly. And they often use coenzymes, many of which are just derivatives of the vitamins you eat in your diet, to help spark the reaction.
And we measure the speed and efficiency of these enzymes using Michaelis-Menten kinetics. Two variables are incredibly important here, Vmax and creatinine.
Let's use an analogy for this because the textbook definitions can be pretty dense.
Good idea.
Imagine an enzyme as a worker on a factory assembly line, putting soles on shoes. Vmax is the absolute maximum velocity. It's how many shoes the worker can process when the conveyor belt is running at full speed and they never have to wait for a part.
Right. And CAMM, on the other hand, measures affinity. It's a measure of how easily the worker can grab the shoe off the belt.
So a low CAMM means the worker has incredibly sticky hands and grabs the shoes effortlessly.
That factory analogy is perfect. Biochemists graph this out to see how an enzyme performs under different conditions. But a standard graph produces a curve that never quite flattens out, which makes it hard to pinpoint the exact Vmax.
So what do?
They use a Lineweaver-Burke double reciprocal plot. By mathematically flipping the data upside down, they turn that confusing curve into a straight line, which makes it incredibly easy to read the exact Vmax and CAMM.
And seeing that Vmax in a clinical setting is fascinating. Let's look at a patient, Al M., who struggles with severe alcohol use disorder. Normally, alcohol is processed by standard liver enzymes, but chronic drinking triggers the body to build more machinery to handle the toxic load. It induces the transcription of a specific enzyme pathway called meos.
Yes, the meos pathway relies heavily on an enzyme called cytochrome P452E1.
Right. So by building more of these enzymes, his liver increases its overall capacity to oxidize ethanol. But enzymes are bound by their Vmax.
Exactly. Once every single meos enzyme in his liver is saturated with alcohol, they are working as fast as they physically can.
The factory workers are completely maxed out.
Which means if he continues to drink, his liver cannot clear the alcohol any faster. The clearance rate completely flatlines at Vmax. The excess alcohol just builds up in the blood, leading to rapid, severe toxicity.
Oof. But here's where it gets really interesting. Scientists are actually hacking this enzyme system to treat addiction.
Yes, they've created something called abzymes, which are catalytic antibodies.
This is such a brilliant application of transition state theory.
It is. When a chemical reaction happens, the molecule briefly morphs into a highly unstable, high-energy transition state before becoming the final product. Enzymes basically work by stabilizing that awkward transition state.
Right. So scientists take a chemical that perfectly mimics that awkward transition state of cocaine, and they inject it into an animal to generate an immune response.
And the resulting antibodies are specifically shaped to bind to and stabilize that transition state.
Exactly. They've essentially built an artificial enzyme from an antibody. When injected into a patient addicted to cocaine, this abzyme rapidly degrades the drug in their bloodstream before it can even reach the brain. It's a targeted biochemical strike.
It completely neutralizes the threat by speeding up its destruction.
But let me challenge the system for a second.
Sure.
If enzymes exist to constantly speed things up, rushing toward their Vmax, how does the body stop itself from burning through all its fuel at once? Like, if I eat a piece of bread, why doesn't glycolysis just run like a runaway train until all my glucose is entirely gone?
Because the body is a master of self-control, utilizing metabolic regulation. These pathways don't run unchecked. They are tightly regulated, usually at the slowest, rate-limiting step in the assembly line.
Okay, so how do they regulate it?
We see feedback inhibition, where the final product of a pathway physically loops back and binds to the enzyme that started the process, changing its shape and turning it off. The product literally says, "We have enough, shut down the factory."
And the reverse is true too, right? Feed-forward regulation.
Yes. If a toxic precursor starts accumulating because the assembly line is backed up, that precursor will push the pathway forward, forcing the downstream enzymes to work faster to dispose of the buildup. It is a constant dynamic balancing act.
So what does this all mean? We've covered the energy pathways, the bioenergetic rules, the folded protein machines, and the enzyme factories. How does a physician actually peer into this microscopic universe without opening the hood?
They do it by reading the metabolic exhaust left behind in the blood and urine. We call this clinical diagnostics.
Okay, let's look at a common dashboard metric. BUN, or blood urea nitrogen. Urea is a waste product generated when your body breaks down proteins.
Right, exactly. The factory that makes urea is the liver, and the exhaust pipe that filters it out of the body is the kidney.
So if a doctor orders a metabolic panel and the BUN is unusually low, that points to liver failure because the factory is broken and isn't producing the waste.
Right. But if the BUN is dangerously high, it points to kidney failure. The exhaust pipe is blocked, and the waste is backing up into the bloodstream.
Another critical marker we look at is creatinine, which is a constant byproduct of normal skeletal muscle breakdown.
And to measure it, laboratories use a fascinating chemical process called the kinetic Jaffe reaction.
I love the elegance of this test. You take a clear biological fluid, the patient's plasma or urine, and you mix it with picric acid in an alkaline solution.
And when the creatinine reacts with the picric acid, it forms a distinct red-orange color.
Yes. The lab uses a spectrophotometer to measure exactly how intense that color is and how fast it forms. More red-orange equals more creatinine.
And that color change tells a profound clinical story. Consider the case of ANR, a patient suffering from severe anorexia nervosa. If a doctor runs the Jaffe reaction on her blood, they will find an abnormally low creatinine level.
Because her body, trapped in a state of prolonged starvation, has been forced to break down her own skeletal muscle to harvest amino acids just to keep her organs running.
Yes, less muscle mass means less baseline creatinine being produced. The biochemical dashboard directly reflects her physical wasting.
If we connect this to the bigger picture, a simple blood draw gives a physician a literal real-time map of the patient's internal universe.
Exactly. Consider our final clinical case, Cora N., who arrives in the emergency room clutching her chest, suffering from a myocardial infarction.
A heart attack. A blood clot has blocked a coronary artery. The oxygen supply is cut off.
And without oxygen, her heart cells can no longer run the electron transport chain. Oxidative phosphorylation stops.
Wow, okay.
ATP levels plummet, and the cells begin to die, bursting open and leaking their internal contents, including that specific CKMB enzyme we discussed earlier, straight into the blood.
So the doctor reads the CKMB spike and knows exactly what is happening.
Right.
But because we understand the biochemistry, we don't just watch it happen, we intervene. Cora is immediately treated with TPA, tissue plasminogen activator.
And TPA is a recombinant protein medication that hacks the body's own enzymatic system. It converts an inactive dormant protein in the blood, called plasminogen, into its highly active form, plasmin.
And that plasmin acts like a biochemical scalpel. It slices up the fibrin proteins holding the blood clot together, reopening the blood vessel.
The oxygen flows back into the heart, the electron transport chain fires back up, and those cells start generating ATP again.
It is literally biochemistry saving a life in real time.
It is the ultimate application of everything we've discussed today.
It really is. And that brings us to the end of our journey today.
We've covered a lot.
We really have. We've traced the path of a meal turning into ATP currency. We've seen how the DNA blueprint dictates the origami-like folding of the protein machines. We've explored how enzymes act as assembly line workers to speed up these reactions. And we've learned how doctors read the chemical signals of disease in the blood and urine to save lives.
It is a vast, beautifully intricate system that operates entirely out of sight.
All right, medical students and science lovers, here is your review question for today's deep dive. If a patient arrives in the ER with severe chest pain and their blood tests reveal an elevated level of the isozyme CKMB, what is the most likely diagnosis? And based on our discussion, what recombinant enzyme medication might a doctor administer to squeeze the blockage?
Well, the diagnosis is a myocardial infarction or heart attack, and the treatment we discussed was TPA, or tissue plasminogen activator.
Nailed it.
Before we go, I want to leave you with one final thought. We've discussed today how your DNA sequence dictates the exact structure of your proteins. But think about this. If our DNA sequence remains exactly the same throughout our entire lives, how do environmental factors like your diet, chronic stress, or exposure to toxins permanently alter which of those genes are actually turned on or off?
Oh, wow.
The answer lies in an emerging field called epigenetics. And it suggests that your biochemistry is not just a static blueprint, but a living diary of your life experiences.
Living diary. That is incredible. Thank you so much for joining us on this deep dive. If you're listening to this on your commute or during a study break, keep asking questions. The next time you look at a patient, or even just look at your own hands, remember, you aren't just looking at the surface. You now know how to lift the hood and read the invisible engine of life.